So what we're going to be looking at in this particular video is we're going to be covering the types of proteins. For A levels, for Cambridge A levels, you have to know that there are two types of proteins. This is just a big class, by the way. We are dividing proteins into two big types, which are known as globular proteins and fibrous proteins. Now, as an overview, before we talk about globular proteins and fibrous proteins, what I'm going to do is I'm going to draw out two polypeptide chains. You can see a chain on the left, you can see a chain on the right. Each of the circles are just amino acids and they are linked together by peptide bonds. So the chain on the left, I'm just going to call it polypeptide chain A, and chain on the right is called polypeptide chain B. Now, if you notice, I'm putting some of the amino acids in red and I'm also putting some of the amino acids in blue. The red amino acids signify that they are hydrophobic amino acids. If you remember, hydrophobic amino acids basically mean they cannot interact with water because they are nonpolar. And the blue ones, hydrophilic amino acids, can interact with water because these amino acids have polarity. Now, compare the composition of polypeptide A and polypeptide B. If you look at polypeptide A, some of the amino acids in red are hydrophobic and some of the amino acids are hydrophilic. We also notice that the number of hydrophilic amino acids outnumber the hydrophobic amino acids. So there are more blue than red ones in polypeptide A. But in polypeptide B, most of the amino acids are hydrophobic. So you might be thinking, so okay, does it matter? What's the big deal? Why is this such, why do I have to care about this? Does it really matter if chain A has more hydrophilic amino acids and polypeptide chain B has more hydrophobic amino acids? Yes, it does matter, because think about it. If we were to put both the chains in water, what will happen? We notice that polypeptide chain A starts to form a very weird folding, or it starts to curl up and form a kind of ball-like structure. Whereas polypeptide chain B is not even going into the water. Most of the chain are just settling at the top of the water. There is just one amino acid, if you notice, where it's kind of going inwards. And the reason why it's going inwards is because it's the hydrophilic amino acid. So if we were to zoom in on the polypeptide chain A, we notice that the hydrophilic amino acids face outside and interact with water. Meanwhile, the hydrophobic amino acids curl inwards because they want to avoid the water. And because they want to avoid the water, they interact with each other. That is why those hydrophobic amino acids are near each other. And this is good because this causes the chain to become a ball-like or spherical structure. But for polypeptide chain B, the chain generally does not interact with water and it forms a kind of thread-like or fiber-like structure. Therefore, the chain on the left that forms the ball-like shape is a globular protein, and the chain on the right that does not interact with water and forms a thread-like shape is called the fibrous protein. These are the fundamental differences that happens between a globular protein and a fibrous protein. If we were to look at the globular protein in detail, globular proteins have some very important characteristics that you must know. First and foremost, globular protein is a water-soluble protein. The reason why it's a water-soluble protein is because hydrophilic amino acids, the ones I'm highlighting, are able to interact with water. Because they're able to interact with water, they are said to be water-soluble, even though they do have some hydrophobic amino acids. But fundamentally, it has mostly hydrophilic amino acids. That's why it's able to interact with water. It's a spherical or ball-shaped protein. It's not circular. You cannot put the word circular in the exam because the protein has a three-dimensional shape. And circular, it's not three-dimensional. So you have to say ball-shaped or spherical. And most of the time, globular proteins are involved in chemical reactions. Now, what do I mean by they're involved in chemical reactions? Some globular proteins can be enzymes, which means to say they react with substrates. There is a type of globular protein which is called hemoglobin, which can bind to oxygen, which, by the way, needs to bind to oxygen, okay? And there is also a type of globular protein we are going to see in chapter 11, okay, of immunity, known as antibodies, and they bind to antigens. So these are just some examples of globular proteins that you should know, and we are going to cover all these proteins in the future. So let me just basically show you 3 globular proteins right here. I'm just drawing out these 3 globular proteins, and let's say these globular proteins are all enzymes. Okay, they are all enzymes, and they are all on one side of the screen, and you have substrates on the other side of the screen. Without going into the detail, we just have to know that enzymes have to react with substrates. The problem is the enzyme is on one side of the screen, on the left side, and the substrates are on the right side of the screen. They're on the other side. So will the enzymes be able to react with the substrate? The answer is yes. Because the enzymes are water soluble, they are able to move inside the watery environment and meet up with the substrates. So because the enzymes are water soluble, the globular proteins can easily move. Because they are water soluble, the enzymes can easily move in a watery environment, for example, cytoplasm of the cell, and carry out chemical reactions. Point of the matter here is just basically this. Globular proteins are proteins that are water soluble. They can easily move in a watery environment to do whatever functions they are supposed to do. We're going to look at other examples of globular proteins in the future chapter. But for now, this information is sufficient. And of course, if we were to look at fibrous proteins are not able to be involved in chemical reactions because they cannot move around in the water. So does that mean they are useless? No. In fact, because they are not involved in chemical reactions or they cannot move around in water, that makes them useful in a different way. Now, what do I mean by they are useful in a different way? For example, the water cannot affect them. So if you were to throw water into fibrous proteins, the fibrous proteins are not affected whatsoever. The fibrous proteins will not dissolve in water. They will not disperse in water. In fact, they will hold their structure and they're unaffected whatsoever, which means to say fibrous proteins can have structural functions. What do I mean by structural functions? Structural functions are basically examples of them will be keratin. Keratin is a type of protein that forms nail and hair. Think about it. When you just basically put your hair in water, does your hair just dissolve and wither away? It doesn't, because hair is a fibrous protein. It is unaffected by water directly. Nails too. Just because you dip your fingers in water, does your nail just disperse and just dissolve? It doesn't. Because the keratin in your nails can hold themselves together and they do not interact with water. Other types of structural proteins also include collagen, which can be found in your skin and blood vessels, and you also have elastin, which can be found in your airways, trachea, bronchus, bronchioles, and alveoli, and also your blood vessels too. We're going to look at some of these proteins in other chapters as well. Now, so sometimes students will be like, I still don't understand why this is important. Okay, I'm just going to attempt to give you an analogy. Now, let's imagine you had a block of salts, okay? A block of salt. This is just an analogy, by the way. This is not a protein. But imagine if you had a block of salt and you had a block of wood. Okay, I know wood is supposed to be brown in color. I have no idea why I'm putting it in purple, but yeah, let's go with that. All right. Now, the salt is water soluble, but the wood is water insoluble. So you can imagine that the block of salt is used to represent globular proteins and the block of wood is used to represent fibrous proteins. Okay, it's just a representation. It does not mean it's the real thing. Now, imagine then if I were to build a tower made out of a block of salt. I put a person on the block of salts and I build a tower made out of a block of wood. And I put a person on the block of wood. Now, and imagine then that these two blocks are standing on water. Because the block of salt can interact with water, it will basically start to dissolve in water. It will start to disperse and break apart. Alright, and that's not good. You do not want to be standing on salt, a block of salt, if you are protecting yourself from the water. But the wood, however, is unaffected. The block of wood is unaffected because they are water insoluble. They cannot dissolve in water. Same thing with the fibrous proteins as well. So the globular proteins can dissolve in water, but the fibrous proteins are able to maintain their shape, structure, and integrity in water. So as you can see, the person who was basically standing on the block of salt is now drowning. I hope the person doesn't die. And the person standing on the block of wood is fine. Although why that person is smiling, looking at a person who's drowning, that's another matter altogether. Anyway, so if we were to look at 6 globular proteins on the left, and we also have about 6 fibrous proteins on the right, and imagine then if I were to put them both in water, what will actually happen? Same thing, we've talked about it earlier. Those globular proteins will start to disperse in water. They'll just equally disperse in water because they can move wherever they want to in a watery environment. But the fibrous proteins will never be able to interact with water, so they stay together and interact with each other. So globular proteins can easily move around in water to do metabolic reactions or chemical reactions, but fibrous proteins just stay with each other and they stick to each other so they can perform structural functions. That's basically what we have to understand from this. So what we're going to look at right now is we're going to be looking at an example of a globular protein. We're going to be talking about something known as hemoglobin. Hemoglobin is just one example of the globular protein. We will be talking about hemoglobin more in chapter 8, but we do need to be introduced to what hemoglobin is. One important thing I want to explain to students is, because a lot of students have this confusion, they say that hemoglobin and red blood cells are the same thing. It is not. I want you to understand that hemoglobin is a protein found in red blood cells. Hemoglobin is a protein, red blood cell is a cell. Please do not assume that hemoglobin is a red blood cell. No, it's not, okay? One is a protein, one is a cell. Two different things altogether. Now that that's out of the way, let's look at the structure of hemoglobin. Now, the interesting thing about the structure of hemoglobin is, to make hemoglobin, you need to have 2 polypeptides called alpha globin polypeptides and 2 polypeptides called beta globin polypeptides. The 2 alpha globin polypeptides have the same sequence of amino acids, which means to say they have the same primary structure, and the 2 beta globin polypeptides also have the same sequence of amino acids, which means to say they have the same primary structure. However, an alpha globin polypeptide and a beta globin polypeptide are made up of different sequences of amino acids. Okay, so though they're made up of amino acids, the sequences and types of amino acids are different. Therefore, the alpha globin and beta globin have different primary structures. So, that being said, let's move on to the next part. We notice that the alpha globins will start to form a secondary structure. You can see those alpha helix right there. And the beta globins will also start to form their own secondary structures. Now, this secondary structure is just a representation. This is not exactly what happens in reality, but it helps you understand that. You can see the alpha helix forming in those chains. After they form the secondary structure, each of the chain will fold further to form a tertiary structure. If you remember the previous videos, the reason why a polypeptide chain forms a tertiary structure is due to the interaction of the R groups, and the R groups can form hydrogen bonds, ionic bonds, disulfide bridges, and weak hydrophobic interactions. Now comes one of the more important parts. Each of those tertiary structures that you can see over there starts to interact with each other. So the 4 polypeptide chains are now interacting with each other. Okay, you can see it interacting with each other right there. And if I were to zoom in, the middle may have something known as hydrophobic bonds or hydrophobic interactions. They may have disulfide bridges. They may have ionic bonds. They may also have, like the blue color ones over there, as hydrogen bonds. And this is basically known as a quaternary structure. Okay, but that's not done yet. To make hemoglobin, however, each hemoglobin, if you notice, I'm putting some green color, bright green color circles in there. These bright green color circles are representing something known as a heme group. So each polypeptide chain will have something called a heme group, which is a non-protein. Okay, and this heme group is important because number one, it contains iron, and number two, it binds to oxygen. That's what allows hemoglobin to function. So when we have the polypeptide chain and we add the heme groups to the chain, that is how you make one hemoglobin molecule or one hemoglobin protein. So what we have to understand for this is hemoglobin is actually a quaternary structure. Why do we call it a quaternary structure? Because to make one hemoglobin protein, it is actually consisting of 4 polypeptide chains interacting with each other. The unique thing about hemoglobin is for hemoglobin to function, you need 4 polypeptide chains and you also need 4 heme groups. Without the heme group, the hemoglobin is useless. So the heme group also needs to be there as well. So just going back to the left over here, I'm just going to cover this. So we have the polypeptide chain on the left. They are all just a sequence of amino acids. They are the primary structure and they form the secondary structure. And from the secondary structure, they form tertiary structures. They form the quaternary structure. You add the heme groups and lo and behold, hemoglobin is made. Hemoglobin is an example of a globular protein because it's water soluble and it also has a spherical shape. And they are involved in chemical reactions because the chemical reaction is they have to bind to oxygen and bring the oxygen to our body cells. And we are going to look at that in chapter 8. So we have looked at one example of a globular protein. Now let's look at one example of fibrous proteins. Now, for fibrous protein over here, the most famous example of fibrous protein you have to say is collagen. Collagen is an interesting one. Now, the structure of collagen is made as below. We have a polypeptide chain. I'm just drawing that one in red. Another polypeptide chain in blue in the middle, and another polypeptide chain in green, which is at the bottom. Okay, and each of these polypeptide chain have different primary structures. The reason why they have different primary structure is because, the reason why it's different is because their sequence of amino acids are slightly different from each other. You don't have to know the names of the chains, by the way. But what's interesting over here is the chains will start coiling within themselves to form something known as a collagen molecule or a collagen protein. That is one collagen protein right there. And that one collagen molecule is a quaternary protein. Why is it a quaternary protein? Because it is made up of 3 polypeptide chains. Never ever lose the definition, never lose sight of the definition, by the way. Quaternary proteins basically means proteins made up of 2 or more polypeptide chain. So just like hemoglobin proteins, collagen proteins are also quaternary as well. And the shape of collagen is referred to as triple helix. Why is it triple helix? Because it's made up of 3 polypeptide chains, and helix because they are spiraling within each other or twisting. Now, upon closer inspection of each of the chain, I'm just drawing out each of the chain here in detail. I want you to see that each of the amino acids have different sizes. Because if you remember, in our video on amino acids, I did say that there are 20 different types of amino acids required to build proteins, and each amino acid have different sizes, and what makes the amino acid different is because of their R groups. So if you see the top right corner, there will be a link to the video on amino acids, but I'll also put it in the description if you want to look at the video on amino acids as well. What I also want you to notice here is each of the chain have different types of amino acids and they have varying sizes. Now, but a very interesting thing is there is a pattern. The pattern here is as follows. Every third amino acid in the chain is glycine, which is the smallest amino acid out of the 20 amino acids. Glycine is referred to as the smallest amino acid. You have to know this. This is very important. And glycine is found in every third position of each chain. Whether it's the chain at the top, chain in the middle, or chain at the bottom, glycine is in position number 3, position number 6, position number 9, and position number 12. I'm also drawing out the glycine molecule here just to show you. You have the carboxylic acid group, you have the amine group, and in purple, I'm drawing out the hydrogen, which is the R group. That's why glycine is the smallest amino acid, because its R group just consists of a hydrogen. Now, you might be thinking, so what if it's glycine at every third position or at every regular intervals? The reason is because where there is glycine, it allows the chain to become closer to each other. And when the chain comes closer to each other, that's what allows the polypeptide chain to coil within each other. So because glycine is quite small, the chains can come closer. And where I'm highlighting is where the twisting happens. That's how it allows the 3 polypeptide chain to tightly coil around each other to form something known as the triple helix. Okay, and it does not easily unravel. And lo and behold, this is how you get a collagen molecule, because a collagen molecule is made up of 3 polypeptide chains tightly coiling around each other. And the tight coils are only possible because of the size of glycine. Because glycine is small, it allows the chain to be closer to each other and twist around each other. That's the significance of glycine in this moment. So we know that one collagen molecule is just basically made up of a triple helix of tightly coiled together polypeptide chains. That's good. What happens if I just basically start taking many collagen molecules? As you can see here, I'm just basically taking many collagen molecules, and I'm starting to draw this orange color lines between the collagen molecules. Those orange color highlighted areas are known as crosslinks of covalent bonds holding each collagen molecule in a staggered manner. Okay, in a staggered manner. Now, students will get very confused with this. What does it mean by a staggered manner? A staggered manner just basically means each collagen is attached to another collagen and they form a kind of stair-like fashion. I'm just drawing it on the right-hand side. You can see how it just forms a kind of, it looks like stairs. It looks like many stairs just bundled up with each other. That's what it's called, staggered. All right. Now, again, is this really important to know? It is, because allowing one collagen molecule to be linked to another collagen molecule through these covalent bonds allow us to form something known as a collagen fibril. So I'm just going to show you a comparison between staggered crosslinks and also straight crosslinks in this case. As you can see, the staggered crosslinks, they are forming a kind of like stair-like pattern. They look like stairs, but the straight crosslinks are just basically it's straight. Okay, so you have the collagen fibril, which have straight crosslinks, and you have collagen fibrils that are staggered crosslinks. Now imagine if I were to exert a force onto both fibrils. If I were to put a force between both fibrils, look at the collagen fibril on the right. What happens is, the highlighted areas, there is a weak spot in the middle, and that's not good to have. Because what happens if you have a weak spot is the collagen molecule or the collagen fibril will just basically, the collagen fibril will just break apart because of those weak spots. But because you have staggered, because when it's staggered, there are no weak spots. The weak spots do not run along this fibril, so there is no proper weak spot. So when you exert a force in the left cellulose fibril, it bends, but it does not break. That is why staggered crosslinks are very important in a cellulose fibril. That's the most important thing we have to know over here. So in this situation over here, we have one collagen molecule, and if we have many collagen molecules forming a crosslink in a staggered way, that's when we get something called a collagen fibril. I'm just going to color it in purple, okay, just to show it over here. That is one collagen fibril. A collagen fibril is made up of many collagen molecules linked together by covalent bonds. And if I have many collagen fibrils joining together, that's when we get something called collagen fiber. And you may have thought, hey, wait a second, this looks a bit like cellulose in carbohydrates. And you are not going to be wrong. You are not wrong, by the way. Collagen fibers, just like cellulose, have a very high tensile strength. okay? The high tensile strength means they will not be easily broken down when you try to push, pull, or stretch them. This is why it's important to have collagen in places where it is subjected to a lot of pressure. For example, the wall of arteries, your tendons, which are connecting muscles to bone, and also your skin, okay? You can pull your skin in many directions and it just doesn't give way because of the collagen in that situation. And the main difference is collagen is actually quite flexible, all right? They're quite flexible. They're unlike cellulose. Cellulose are quite rigid, but the difference is collagen, which is a protein, is quite flexible. So in this diagram over here, you can see, I know it's zoomed out a little bit, so I hope you can actually see this. You can actually see on the left hand side, we have one collagen molecule, which is just a protein. And when we have many collagen molecules linked together in a staggered way by forming crosslinks, they form something called collagen fibril. But when you have many collagen fibril grouped up together, you get something known as a collagen fiber. So with that, we are actually done with the comparison between globular proteins and also fibrous proteins.